Welcome back, everybody. Let's get started with the afternoon session. And the next lecture will be given by 5 students and postdocs who won the Abstract Award. And this award is based on our ratings of the abstracts, so not the posters presented here, but just the abstracts anonymously. And as every year, we've received many fantastic abstracts, so it was difficult to come up with a short list, and so that's why we had, maybe I should show you the actual names, but we had actually 9 winners this year, but 4 of those 9 have won the award previously, and so they will not give a presentation today, but they will present the poster on their work. And the 5 people who have not won it previously, they will give a short presentation of 12 minutes, followed by 3 minutes of discussion. And so there will be just maybe 1 or 2 questions per talk. And so I'll try to keep very strict limit on the presentation time and the question time as well. The first person to present is Valentina Bruno, and she will present a study, the functional role of ventral premotor cortex in motor monitoring and fMRI study. Thanks, and congratulations.
Thank you. So, thanks to the organizers for giving me the opportunity to present this research, the functional role of ventral premotor cortex in motor monitoring, an fMRI study during movement execution and mechanic limb immobilization. One of the main topics of the research group I belong is the motor awareness, that is the ability to monitor our actions and to predict their consequences. The pathological model that allows us to investigate the motor awareness is the anosognosia for hemiplegia. After a brain injury, typically involving the right hemisphere, patients with anosognosia show a paralysis of the contralateral part of the body, but they are not aware of their motor deficits, and they claim can still move the left plegic arm. If the examiner asks the patients to move the left plegic arm, the arm remains still on the table, but the patients firmly report to have done the requested movement, showing a completely altered motor awareness. It has been proposed a model, an anatomical model of the anosognosia for hemiplegia, based on lesion and behavioral data. In these studies, based on brain-damaged patients with a contralateral paralysis, it has been shown that in patients with anosognosia for hemiplegia, there is a balance between spared and damaged areas. The supplementary motor area, that is the part of the circuit where intentions and motor programs born, is shown to be spared in the majority of the cases of anosognosia for hemiplegia, while the premotor cortex is shown to be selectively damaged in this kind of patients. The premotor cortex should have the role of comparator system, and this should be involved in the motor monitoring of action. The model proposes that the comparator system should compare the efference copy built on motor programs with the sensory consequences that comes from the action. If the comparator is damaged, such as in patients with anosognosia for hemiplegia, the system loses the ability to compare the motor programs with the sensory feedbacks, and this produces in the patients with anosognosia the false belief to be able to perform movement that they intentionally programmed. In other words, these patients are aware of the movement they intend to perform instead of the movement they actually perform. The role of premotor cortex in motor monitoring was demonstrated recently in a study by Bolognini and co-workers on healthy subjects. They used cathodic direct current stimulation in order to create a virtual lesion of the premotor cortex, and they asked participants to perform a motor monitoring task. The results show that the participants' ability to evaluate their motor performance was significantly impaired after cathodic direct current stimulation over premotor cortex, making them unconfident about their judgments. But what is the aim of these studies? we sought for evidence of a comparison between efferent output and afferent input of movement in premotor cortex. To this aim, we contrasted conditions in which subjects were free to execute a hand motor task with conditions in which the movement was prevented by a mechanic limb immobilization. We predict that in free condition, subjects and also the areas involved in the motor monitoring should not find any incongruence between the intended and executed movement. In blood condition, we predict that the areas involved in the motor monitoring system should inform the system about the incongruence between the intended but not executed movement because of the mobilization. Therefore, we predict a stronger activity of premotor cortex. Additionally, we ask whether the activity of the premotor cortex can be modulated according to the duration of the immobilization. 20 healthy subjects were recruited and they underwent 4 fMRI scanning sessions. And so the experiment included 4 time points. The first day, subject had to perform a hand motor task that I'm going to explain in the next slide with both hands free. At the end of the scan, the left hand was blocked with a cast and they performed the task with the left hand blocked and the right hand free. After 7 days of immobilization, the second day, subject had to perform the task with the left hand blocked and the right hand free. At the end of the scan, the cast was removed, and they finally performed the task with both hands free. During the fMRI scan, participants had to move or try to move in blocked condition their left hand and right hand alternately. In rest condition, they had to relax without performing any movement. The movement consisted in a flexion and extension of the 5 fingers conjointly. The stimuli were visual and represented 2 hands. Both hands were white in rest condition. When the right hand turned red and the left hand remained white, the subject had to perform the task with the right hand. Vice versa, when the subject had to move the left hand. The task was performed using a design block with 12 seconds of rest and 12 seconds of active movement. All the analyses were performed using SPM8. We took advantage of the meta-analytic software NeuroSynth in order to identify the region of interest that are commonly implicated in right-hand movements. And so we identified left M1, left SMA, left ventral premotor cortex, and left dorsal premotor cortex. Specular rows were used for the left-hand movements. We extracted the brain activity, the beta values of each region of interest, and we entered those data in a 2x2x2 ANOVA with 3 two-levels within subjects factors. Side, right, left hand, including the contralateral hemisphere activity, and right hand, including the contralateral hemisphere activity. Time, day 1 and day 2, and cast. Free session when both hands were free, and blocked session when the left hand was blocked and the right hand remained free. I would stress that only the left hand was immobilized, while the right hand remained always free. We found significant effects only when considering M1 activity and ventral premotor cortex activity. In particular, we found a significant side per cast interaction both for M1 activity and for VPM ventral premotor cortex activity. For the right control hand in blue, we didn't find a significant difference between free and blocked condition, both for M1 and for ventral premotor cortex activity. For the left manipulated hand in orange, we can see an opposite trend of activity between the M1 activity and the ventral premotor cortex. In the M1 activity significantly decreased in blocked with respect to free condition, while the ventral premotor cortex activity significantly increased in blocked with respect to free condition. For the ventral premotor cortex activity, postdoc comparisons show also a significant difference between left hand and right hand in blocked session. While for the M1 activity we didn't find a significant three-way interaction, we found the significant side per time per cast interaction for ventral premotor cortex activity. Here we plotted the bold signal change normalized on the 4 time points of each subject. You can see in orange, you can see the average of the activity in orange and in blue, and the results of single subjects in gray. We found a significant increase of ventral premotor cortex activity at time point 2 with respect to time point 1. For the left manipulated end in orange, obviously. For the right control end in blue, it's possible to see an opposite trend of activity with higher beta values at time point 1 with respect to all the other time points, but it didn't reach the significance level. So, according to our hypothesis, when the left hand movement are prevented by a cast, by a mechanic limb immobilization, we found a significant increase of premotor cortex, and in particular of ventral premotor cortex in premotor cortex, suggesting this area involvement in detecting the mismatch between intended and executed movement. It suggests that when afferent and efferent signals are non-congruent as when they are prevented by a cast, as when the movement is prevented by a cast, the activity of premotor cortex significantly increased. It is worth noting that this motor monitoring function of premotor cortex can be dissociated by a pure motor function, such as that present in M1 with lower beta values when the cast was placed on the end. Additionally, answering to the question number 2 about the modulation of the activity of premotor cortex according to the duration of the immobilization, we found that after 1 week, when the system learns that no movement can be executed with the left hand and changes its prediction about the sensory consequences of the movement, the system no more detects the incongruence. The present findings provide a compelling evidence for the proposed anatomoclinical role of premotor cortex in comparator system. For the future, we are planning to make further analysis in order to study the connectivity between these areas in order to describe with a more complex model this motor monitoring system. I would like to thank my research group, the Samba research group, and also my colleagues of the University of Turin, and also the colleagues of the University of Pisa. Thanks also to the SEER grant for funding us, and thank you all for your attention.
Thank you. We have one question here.
Thank you. Congratulations for your work. I'm just wondering if you did you try to test the limb mobilization on plasticity according to other neuropsychological or neurophysiological measures?
Yes, thank you for the question. Yes, we unfortunately not hold the sample of our experiment, but part of the participants of our experiment underwent also 2 sessions of transcranial magnetic stimulation in order to study, in order to investigate possible changes in corticospinal excitability after the immobilization. And we found, as demonstrated in previous studies, that after the immobilization, the participants show a decrease in the corticospinal excitability. In particular, we found significant increase of the resting motor threshold. And so, according to our studies, yes, we can say that we have found a measure that, that explains the plasticity phenomena that occur after the immobilization. Thank you.
So if I understood correctly, you didn't see any difference, the difference you found in ventropremotor cortex was between the first and second time point, but there was no difference neither in M1 or ventropremotor cortex between T2 and T3, which was the long-term immobilization phase, right? So I was wondering, did you find any areas, I found that a bit surprising, so did you find any areas where you did find a difference, and if not, do you think that the period of immobilization induced changes in connectivity that you then might see?
So, unfortunately, we didn't find a significant three-way interaction for M1 activity, yes, but maybe I have the graph. But we found the activity of the M1 activity significantly, M1 activity, no. Okay, it's this one, the three-way interaction of the M1 activity. Yes, for the left immobilized, the manipulated end in orange, we found the interaction is not significant, but we looked also the postdoc comparison, and postdoc comparison showed that there is a significant decrease between, a significant difference between time point 1 and time point 3, because obviously for the learning of the paralysis, yes, sure. But at the end, there is a significant difference also between time point 3 and time point 1. So the system, it seems that the system needs more activity to move the left immobilized end at time point 4. So, yes.
Thank you very much.
Thank you.
So the next presenter will be Daria Proklova.
Okay. Can you hear me? Okay, hello everyone. I'm going to present our project called Decoding Object Shape and Object Category with MEG that we did here in the University of Trento. Okay, as we know from the literature and from yesterday's talks, there is this animate distinction in ventral temporal cortex, with animate objects activating more lateral regions and inanimate objects activating more medial regions. However, if we look closely at the different categories, we can see that some categories, such as bodies, tend to also have typical shape that distinguishes them from the other categories. And same goes for faces, four-legged mammals, and also for inanimate objects. So tools tend to have elongated shape. Cars and houses also have typical shapes. So it raises the question whether this categorical distinction that we observe is really due to category, or it might be partially explained by some of the shape features that define these categories. So this is the question that we addressed in an fMRI study. So to disentangle animacy and shape, we came up with a stimulus set in which we had animate objects, this is a snake, and inanimate objects, such as rope, that were different in category but had very similar shape. And we did this for 4 different shape subsets. So within each shape subset, we have 2 animals and 2 non-animate objects that are matched for overall shape features. So because we selected stimuli somehow intuitively and arbitrarily, we wanted to measure the visual dissimilarity to quantify it. So for this, we conducted a separate behavioral experiment in which participants were doing a visual search task. This was adapted from Mohan and Arun. So on every trial, they were looking for a target among the identical distractors. So, and we did this for each target distractor pair from our stimulus set. So in this case, it's pretty easy to find a target. And the task was just to find, to respond if it's to the left or to the right of the central line. So we measured the reaction times in this task for each pair of target and distractor and created a visual dissimilarity matrix in which each element shows the inverse reaction time for each pair of objects. So for example, high values correspond to short reaction times, which corresponds to high visual dissimilarity. So these objects were visually dissimilar, so it was easy for participants to find them. And on the contrary, if there is a low dissimilarity value, it corresponds to high visual similarity, so low dissimilarity. So participants took longer to find these two objects among each other because they are so visually similar. So then we also visualize this matrix using multidimensional scaling. And as you can see, our 4 initial shape clusters really do cluster together. So objects from the shapes were indeed visually similar. So then we went on to do fMRI experiment. This is the overview of the experiment. So participants were doing a simple one-back task when they saw 2 images of the same object twice in a row. Okay? And this is the schematic of the analysis that we did. So for each pair of objects and for each region in the brain, we measured the activity patterns, and then we measured 1-R correlation between these stimuli and created a neural dissimilarity matrix. And then, so this category dissimilarity. Then we modeled this neural dissimilarity matrix as a combination of category dissimilarity and visual dissimilarity. So here, the visual dissimilarity matrix comes from the behavioral experiment that I just showed you, whereas category dissimilarity is just a binary predictor, so it has maximum dissimilarity for objects from different categories and 0 for if objects are from the same category. Okay? And this is the results of the fMRI experiment. So these are the 10 slices covering the ventral temporal cortex. And in red, you can see areas in which neural dissimilarity was predicted very well by the visual dissimilarity. So we can see that it's very prominent in the early visual areas and also extends to more anterior portions. And in green is the category information. So we don't see it in the early areas, but we see it more in the anterior portions of VTC. And then in yellow is the overlap between these two. So from this experiment, we concluded that both shape and animacy are represented in ventral temporal cortex, and we cannot explain this categorical distinction simply by visual features. So then we wanted to do the MEG experiment with the stimuli. So the questions we were interested in is, when can we decode category and shape information, and can we disentangle them? So we know from previous studies that category information, and specifically information about animacy, can be successfully decoded from MEG signal around 100 milliseconds with a peak at 157. But the question still remains if we can replicate this with stimuli such as ours, where we also control for shape. So this is the MEG experiment design. So it's very similar to fMRI, but we had an oddball detection task. So when participants saw the hammer, they had to press the button. Okay. So this is the quick overview of the decoding. I'm not going to go into much detail. So basically, we measured the MEG sensor patterns for at each trial and for each time point. And then for each pair of conditions, so here I and J, we train the classifier to discriminate between this pair of conditions, and then we test on some remaining portion of data. And we did this for each pairs of our conditions and created the decoding accuracy matrix. So here, each element of the matrix shows you the average decoding accuracy for this pair of conditions. Okay? And this gave us the measure of neural dissimilarity of our stimuli. And then we could do the same analysis approach as we used in the fMRI study. So we took this neural dissimilarity, which contains the decoding accuracy information, and we modeled it as a combination of category dissimilarity and visual dissimilarity, still the same that we used in the previous experiment. Okay, and the interesting part is that we could do this for each time point. So we could see at which time point the contributions of category and visual dissimilarity are present. Okay, so these are the results. So here is the contribution of visual dissimilarity to the neural dissimilarity. And we can see that starting from around 80 milliseconds and peaking around 100 milliseconds, the visual similarity contributes a lot to the MEG signal. However, when we looked at the independent category contribution, we found nothing. So after regressing out this visual influence, we couldn't find residual category information. But then, as you remember, the task in this study was just to detect the oddball, so press the button when there's a hammer. So we thought maybe our task didn't tell participants to process the other stimuli. So maybe they were just doing hammer, not hammer, hammer, not hammer. So we wanted to replicate our fMRI experiment design. So we repeated the experiment with 15 more participants, this time using the same task that we had in fMRI. So one back task on the object type. So participants hopefully thought a bit more about what the objects are. Okay, and this is the result. So again, we have a very strong influence of visual dissimilarity, again around the same time, 100 milliseconds. However, still no category contribution. So from this, we concluded that MEG as a sensor patterns evoked by the visually presented stimuli predominantly reflect the visual object properties. And given our previous fMRI results, we suggest that MEG may be less sensitive to category information compared to fMRI. Okay, and I want to thank my co-authors, our lab, Cosmo MVP, and Attend Grant. Thank you.
That was great. I wanted to get you to clarify for us how you're thinking about visual similarity, because it seems like you could be thinking about the similarity of the pictures you have or the exemplars that you've chosen, or you might also be thinking about visual similarity as the similarity of kind of the prototype exemplar of that category or of all exemplars of that category. And it seems like the further you go down that road, the harder it's going to be to separate category effects from visual similarity effects. So I guess I wasn't clear where you were trying to operationalize or think about visual similarity as distinct from category similarity. Does that make sense?
I guess. So in this experiment, we really focused on the visual dissimilarity as measured in this visual search task. So we hope that reflects not only the overall shape similarity, but also other maybe contributions of texture or like a contribution of the outline of the stimuli. And also we did 2 other visual search experiments. I can maybe show you. So we repeated the visual search experiment, also using this texture patches of a stimuli as the targets and distractors, or the outline of the stimuli. And we did the same task, and this gave us more dissimilarity matrices that were capturing some other aspects of visual dissimilarity for a stimuli. So we also we looked at all of these. But of course, if you continue matching all the features, then at some point there will be some features that really define the category. So of course, we cannot completely separate these two.
Right. This is really cool. I guess what I'm wondering is if you randomly chose an exemplar of a bird, I don't know what kind of bird that is, but let's say whatever it was perched on the limb of a tree. So it's not like this, right? Or an airplane that's sitting in a side view on the runway, not up in the sky. Your measures would be completely different. Of course. Right?
Yeah.
So it really is about the image that you're after.
This is specific to the stimuli that we selected.
Okay. Thank you.
So thank you. You were unable to classify like using MEG, and you stayed in the sensor space. Did you try to go in source space?
Not yet. This will be the next step. So now we just took all the sensors and tried classifying them.
I have a question in the back. First in the back.
Some time ago, we did an experiment very much like yours in fMRI, not in MEG, where the task was a one-back task, but we had 2 conditions. In one, it was visual similarity. In another, it was semantic similarity. And just like you, when you ask people to focus on visual similarity, you don't get category effects. But when you ask people to focus on semantic similarity, guess what? You get semantic effects. And you get category effects. Okay. So I think it very much depends on what the expectations of the experiment, of the participants are when they perform these sorts of tasks. We didn't expect this at all. We thought when we were asking people to focus on visual similarity that they would implicitly pick up the semantics, but they didn't.
Yeah, this didn't happen in our study, and even when we used another task that subjects really had to think what the object is. it still didn't improve the performance.
Okay, thank you very much.
Okay.
And our next speaker will be Kamila Jozwik.
So I would like to thank the organizers for giving me the opportunity to give this talk. And the title of my talk is Visual Features versus Categories, Explaining Object Representations in Primate IT and Deep Neural Networks with Weighted Representational Modeling. So for object representations in primate inferior temporal cortex, there's evidence for the importance of visual features of intermediate complexity, such as patch, shape, color, and texture. And also other studies indicated that semantic categories and category membership are important for the primate IT representation. However, there's little really studies, and actually the previous talk was relating to this question as well, comparing really the contribution of visual features and semantic categories. So we ask the question, to what extent do features and categories explain the IT object representations in primate IT and in deep neural networks? So we have 15 subjects that generated labels for a set of 96 images in terms of the visual features and semantic categories. So for example, for this image, the visual features were eye, skin, elongated, and semantic categories were, for example, organism, female, face. And we had the set of 96 images from Kriegerskotte and Chiani, and you can see that they span across faces, animals, body parts, tools, scenes. And we basically built our model based on the images labels. So this is just part of the matrix for the feature-based model. You can see that the rows represent images and the columns represent the labels. And if a given label applies to a given image, then it's indicated in a black rectangle. So for example, an ear applies to a monkey face. So if we zoom out and look at the full feature-based model, and then we end up with 120 features, whereas for the categorical model, we end up with 114 categories for the set of 96 images. And now one option is to basically take each of the feature and assume that each of the feature or each of the category contribute to the representation to the similar extent. But we know that's not really the case, that some of the features or categories may be more important for the representations. That's why we wanted to optimally weight each feature and each category to predict our data. And here the green circles represent the weights schematically. And we use cross-validated non-negative least squares, and we ended up with a model prediction and some unexplained variance residuals given our data. And this approach is powerful because of the two reasons. One, it improves the model prediction, and the other is that you can interpret these weights actually. So you can think and see what really are the features and categories that they explain the representation. So let's look at the model performance for human IT. So we correlated our models with human IT data. We estimated the noise ceiling, which tells us, giving the noise in the data, what would be really the performance of a true model. So feature-based models significantly correlated with human IT data, same as for categorical model, but there was no significant difference between these two models' performance. And now let's see what happens if we combine these two models. One option is that categories and features explain unique variance. If you combine them, then the performance will increase. Second option is that they are highly correlated, and actually combining them won't improve the performance. And that's what we see actually. And so we published this study also looking at some behavioral measures, but then we were thinking, okay, so what happens if we look at the models of human IT? How well these models would actually fit to two best, arguably, models of human IT, which basically is monkey IT and deep neural networks. So for the monkey IT, again, we correlated our models. We observed that feature-based model significantly correlates. Same with categorical model, but there's no significant difference in terms of how well they explain the data. And the combined model does not improve model performance. So for primate IT, if we think about explanatory model variance and non-explanatory model variance, features are correlated with categories, and categories are correlated with features. And that's why a feature-based model and a categorical model explain the equal variance. But then we don't really know what features and levels of categorization are most important for the representations. So we wanted to dissect our models and, in terms of the visual features, categories, or visual features classes, and parts, colors, textures, and shapes. So basically, the feature classes that were shown to be important for IT representation. For semantic categories, we divided them in basic categories, intermediate level, and high level categories. For example, for this image, the visual features, the parts would be lake, colors, beige, textures, woolly, shapes, rounded, and semantic categories of basic level would be sheep, intermediate animal, and high level organism. And then let's look at our dissected model feeds to human IT. These are the full models, and the dissected categories will be shown in green, and dissected features in purple. So intermediate level categories, they significantly correlated with human IT, same with high level categories and basic level categories. However, basic level categories perform significantly worse than the previous two models. For the feature-based model, parts dominated the representation because the colors, texture, and shape were not significantly explaining IT data. So let's look now at the pairwise comparison between these models. And when we look at the first 6 models, actually there's no difference in terms of how well they explain IT. That means that intermediate level categories and high level categories are the most important for categorical representation, and parts are most important for the feature-based representation. And let's look at the monkey IT model fits. This is what I showed you for models. And again, high-level categories and intermediate-level categories are significantly better than basic-level categories. And for the feature-based model, parts and color explain the representation. However, the color is significantly worse than parts. And features and shape models didn't really significantly correlate with the data. And in terms of pairwise comparisons, again, actually, there was no difference between the first 6 models, meaning really that high-level categories and intermediate-level categories and parts are dominating representation. So now let's go to deep neural networks. So we know that they reach human-level performance of different tasks, for example, image classification tasks. And also we know that they are actually explaining IT representation quite well. So Kamila Jozwik from our lab correlated each of the layers from Krzyzewski network with the human IT. So layer 1 significantly correlated, but didn't explain much data. Intermediate level, they explained more data, and the high level explained the majority of the data, however, not everything. But still, that's great that they explain a lot, but we don't know really what is represented in these layers. So we wanted to know what layers of a deep neural network are features and categories mapped to. So that's why we basically fitted our models to each of the layers. And always the sequence will be feature-based model, categorical, and combined. So let's look at the layer 1. Here, only feature-based models significantly correlate with the data. And in terms of the dissected models, they are parts and colors that dominate the representation. For the intermediate layers, both categorical and feature-based models significantly correlate with the data, and high-level and intermediate-level categories explain the majority of the categorical model, whereas for feature-based model, it depends on the layer. So for layer 2, it's color and parts. Layer 3, it is color, part, and shape. Layer 4, it is texture and parts. And layer 5, texture and parts again. Whereas for the high layers of the network, again, feature-based model and categorical explain a lot of variance, and they explain more variance than the previous layers. And parts and color, but parts are significantly higher than color, dominate the feature-based representation, and again, high and intermediate level categories. So from that, this is really consistent with what Said showed, and it makes sense that layer 1 doesn't correlate well with IT because it doesn't have categorical representation. And intermediate layers, they do have representations of categories and features, but on a lower level than the late layers. And that's why the late layers basically explain the representation the best, because the categorical and feature-based representation is the highest there. So to sum up, both feature-based and categorical models explain the IT object representation significantly and to a similar extent in human and monkey. Object paths and categories at high levels and intermediate levels of abstraction were most important for explaining IT object representation, and early layers of the deep neural network were dominated by features, whereas both categories and features dominated later layers. So we can conclude that primate IT and deep neural networks use these visual features, mostly object paths, that are associated with high-level and intermediate-level categories are kind of stepping stones towards semantics. So thank you very much for listening, and I'm happy to take questions.
Hey, very interesting stuff. I had a question about the coding of the variables. It sounded like you did this manually by saying this is a part, this is color, that kind of thing. Did you guys think about doing any data driven approaches to that, like maybe clustering on the mappings across the images, or using a PCA space to try and pull out the components that actually were important as opposed to just labeling them yourselves?
Yeah, so kind of there are two elements to this question. First, in terms of how we label these images. So I didn't go into that in details, but basically we got 16 subjects that went through all of these images and said, what are the visual features in these 4 sub-levels, and what are the categories in these 3 different levels of categorization. Then from that, we took 20% of consensus, and another set of 8 subjects validated these features. So basically, look at every feature label, and then for each of the image, and then said, oh, this is true or it's not true for this given image. From that, we took 80% of the consensus, and then we removed the highly correlated predictors based on the iteration. So basically, removing everything that is more correlated more than 0.9. And basically, from that, we constructed the models. And that's true that these visual features I'm talking about are the label ones. So actually, I think if we took the computational models, as you are suggesting, and extract the color information, the shape information, and path information, actually probably these would significantly correlate with IT. That would be my bet. But this is what we want to do as well. For now, we wanted to more understand what is kind of the limit of labeling different features and categories, and basically look at the weights, what is actually most important, what specific category label or what specific feature label from what class is most important for representation. But that's definitely on a plan that we want to look at computational models from these images.
I also have a question. So I was a bit confused about the distinction between the features and the category labels. So when you say, for example, sheep or animal, those are associated with features. And maybe that's actually your conclusion, that in the end, you don't need to have those higher level labels. You can have the part-based labels, and that can explain the representations in IT as well as in deep neural networks. Am I correct in this?
Yeah, you are totally correct. And yeah, that's exactly what's saying, that what we were trying to say, that visual features are correlated of categories and the other way around, and only the visual features that are informative of categories, so some features that are informative of sheep, then actually these will be selected and represented in IT because it's useful to have them for also categorization. And it's very, it's hard to distinguish some of the features and categories. But basically the instruction to the participants was, please name the category that this object belongs to, and for the features, please name the visual features that this object has. So that was kind of free labeling task. And then we, of course, corrected if somebody made a mistake and, like put a category label for a feature. But I totally agree that it's quite difficult to really distinguish. And from this conference, we also see that what is the understanding of features also differs. And it's not easy to define. Yeah.
So what would you expect in the neural network layers for the stimuli of Daria, that the previous talk? Would you expect there to be any categorical organization, or?
The representation of features and categories would be similar for a different set of stimuli. That's what.
So when you try to come up with stimuli that are somewhat matched in terms of visual similarity, would that would then the categorical model fall apart?
I'm not sure I'm getting your question. So if basically you are saying that you are trying to match the stimuli to each of the layers.
Yeah, in the same way that you did. So if you have, or the same way as the paper that you cited did. So if you have a stimulus set in which features and categories are correlated with each other,
yes,
then a categorical model can explain the representational space, or you can at least train it to represent it. But if you decorrelate those,
yes, so I think that you can distinguish them if you have the optimal stimuli set. And actually, this is kind of a different question, because we took the most natural things. Well, they're not actually the most natural, because you can go for scenes and you can go for movies and they're even more natural. But like to kind of it was quite natural for our kind of level of understanding. And for example, I did similar study in terms of rating similarity between faces using Basel's face-based model. And again, there, face, like shape, texture models, neural networks, everything is correlated. And what we are doing, we're actually trying to iteratively pick the set of stimuli that would distinguish these models from the Basel face space, to distinguish, you know, texture from shape and so on. But it's kind of a different question, and it is very well defined and kind of controllable models, whereas this kind of images that are natural, it's much harder to actually come up with this stimuli set that you can control for everything.
Okay, thank you.
Thank you.
Thanks. Next speaker is Stefania Benetti.
Okay, thank you. Just would like to join my the previous speakers and really thanks the scientific organizers. It's really like after chasing death around Italy for almost 2 years, it's a really great pleasure to present this work to you all. So I think most of you are familiar with the notion of cross-modal plasticity and cross-modal recruitment in the blind individuals. We've heard also about this yesterday. So I'd like to move your attention to a different sensory deprivation, which is deafness. And there are like less studies on cross-modal plasticity in the deaf compared to the blind, and I think I can guess why. And with this population, actually, it's very difficult, you know, to carry out research in the scanner and communicate with them when they are in the scanner. But I think the studies are like providing us evidence that cross-modal organization of the auditory regions can also occur. And the most consistent finding is actually, that the auditory cortices in the deaf people can be recruited by visual and somatosensory spatial processing, so brain functions that are mostly like within the dorsal processing pathway. This is a very influential one, actually the first study showing that the right auditory cortex in the deaf individuals responds to visual motion. Still, there are like a number of questions that remains open and relates mostly to the principles and to the mechanism that might be driven cross-modal recruitment of auditory cortices, and with the exceptions of very few animal models, and mostly from Stephen Lomberg group, who showed that there is actually a functional selective response of auditory cortices in deaf cats to visual localization and to movement detections. The evidence in support of a functional selectivity of cross-modal recruitment in the deaf is still scarce. So in a lateral and ventral portion of the superior temporal gyrus, I think you also might be familiar with this, lies these regions that is very sensitive to human vocal information. It's the so-called temporal voice area. This is the seminal work by Bellan and colleagues. So this region has also proved to functionally interact to more posterior visual face areas during processing of related to person and to person recognitions. And there is also recent evidence that there might be an actual like direct anatomical connection in between these two regions. So we believe that this made the temporal voice area in the auditory cortex a very good candidate to show cross-modal recruitment in the lack of acoustic information. So the first aim of this investigation was therefore to see whether we could detect cross-modal recruitment that was functionally selective in the ventral auditory stream. And our expectation was that temporal voice area might selectively respond to faces compared to other visual stimuli that were not relevant for person recognition and person information processing. And the second aim was to look at whether particular pattern of connectivity might actually be associated with potential cross-modal recruitment in these regions. Beg your pardon. Okay, so this purpose, we combine imaging, neuroimaging, and behavioral measures of face processing, and we included 3 different groups, a group of early deaf individuals. They were all profoundly severely deaf, and some of them would use sign language, some others would be educated on using like speech reading and actual speech. And then we matched a group of hearing individuals that were able to use sign language at the same proficiency level, and a group of hearing controls. And we ran 2 very simple functional localizers. So a voice localizers that was run in the fMRI only in the hearing control group, and this was to functionally localize the temporal voice area in this group. And a face localizer in which we contrasted very static faces with neutral expression to houses, and after having matched them for low-level visual features. And we ran these first in the 3 groups in the fMRI, and also we rerun the same face localizer in the early deaf and the hearing controls in the MEG. So the task for both functional localizer was just an end-back task, just to respond where they will see 2 same faces or houses, or we will hear 2 same voices or objects, and so on. We also obtained a composite measure of the ability of these individuals to process visual information, and this was done by combining scores on very well-known face recognition task, the Benton, and a delayed face recognition test that was developed ad hoc for this study, and that was administered after the fMRI session. Okay, so analysis, very simple univariate analysis of regional responses showing the selective face network in the 3 groups. And actually, in the, sorry for this, okay, so in the deaf group, we also spotted this cluster of activation in the lateral portion of the superior temporal gyrus going more ventrally in the superior temporal circus. So when we contrast the deaf group with the 2 hearing groups looking for an interaction effect, so we compare them faces versus houses, these regions actually showed to a significant increase of activation in the deaf compared to the other 2 groups. And this was only in the right mid portion of the TVA. So here you see in blue depicted the temporal voice area, and in yellow the region showing a significant differences in between the deaf individuals and the hearing controls. And when we plotted the beta parameters that were extracted from this region in the deaf and from the group maxima for voice response in the control, we saw that this effect was driven by an increased activation of the TVA in response to faces, but not to houses. And also the interaction in between the 3 groups, the interaction effect was significant on these measures. So at the behavioral level, the deaf individuals that are depicted in blue here show that they were better actually performing the face recognition tasks, so they had higher composite measures. This also was true for the hearing signers though. So we asked whether there was a sort of relationship in between what we saw at the neural level and what we were seeing at the behavioral level. So we took the individual measures of strength activity in the TVA, and we correlated them to these individual measures of phase processing, and we found this this trend for positive correlation between the two measures. So this was not seen in the other two groups, so I did not observe the same sort of correlation in the hearing controls or in the hearing signers. So TVA seems to be responding to phases in the deaf. The next question is, when is this happening? So is it happening at the same time of phase discrimination or recognition in the FFA, or is it maybe like more an epiphenomenon? So is it coming afterwards? The different maybe stage of information processing. So we try to answer these questions with the analysis of the MEG data. And so this is a source-based analysis, and it's based on the power. So we just contrasted powers in between condition on the top. So you see again the phase selective network coming up, and the contrast in between deaf and hearing controls at the bottom, and again the interaction effect is significant, and you see that actually MEG was able to locate this effect in a location that is very similar to what we saw in the fMRI. And when we plotted the responses on time scale, and we use region of interest here, so we use the region of interest from the fMRI analysis to look specifically at FFA, which is on the top, and TVA, which is at the bottom. And you can see in gray is depicted the time window that goes from 160 milliseconds to 200 milliseconds. So let's say the time window in which phase information is integrated and recognized. And what you can see is in the early deaf, the response in the FFA and TVA is very similar. We don't have this in the control group. So since that what we were like locating in the right temporal voice area of the deaf, it's happening at the same time that what we would observe in the FFA of the same sample. So last question was about connectivity. So we asked whether it was possible to explain the phase selective response that we saw in the right TVA in terms of some sort of alterations of connectivity. I'm referring to functional connectivity here. So the first thing we did was just like in a data-driven way to look at simple functional correlations of activity between the seed region, that is our TVA, and any other region in the brain. And what we found in the early deaf was indeed an increased and very strong functional correlation between the activity of the seed region and the activity in the visual posterior face areas, but also into a more posterior region that was actually in the occipital and early visual cortex. This is V2, V3, and extending more laterally to EBA. So when we compare these in the deaf and in the other groups, this effect held, but you see here that actually the stronger functional correlation was found in the visual cortex. So functional correlation is nice, but it doesn't tell us much about the directions of interactions, and also it doesn't tell us much about the temporal dynamics of these interactions. So we decided to go further and run some dynamic causal modeling analysis. We built, like in our model space, 3 very simple models that were physiologically plausible to test the hypothesis that the phase selective response in TVA might be driven by a increased forward connectivity from the early visual cortex. And what the model selection told us was that indeed phase selective response in TVA was associated with increased forward connectivity from visual cortex to the auditory cortex during phase processing. So to summarize, we saw phase selective response in the right TVA in the deaf compared to the other groups. So this might actually suggest that functional selectivity is a principle of across model recruitment in this group of deaf individuals. We also find a positive relationship between TVA response and face processing abilities in the same group, so maybe pointing or suggesting some sort of, I wouldn't say causal relationship, but a relationship in between the behavior that we observe in these deaf people and the ability to process face information better than the hearing controls and the neural responses in the right TVA. And last, we also observed an increased forward connectivity from visual cortex to TVA that was supporting the face selective response in this region, suggesting that maybe cross-model recruitment that has this functional selective feature might also be supported by a rearrangement of large-scale connectivity that might happen very early in the development of these individuals. So I just like to thank all the collaborators in the cross-model plasticity, cross-model perception and plasticity group, Olivier Collignan, and I also want to thank two brilliant students, Giuseppe Rapini and Joshua Zonka. They contributed very much to the carrying out of the experiment and the analysis, and Markus also for helping with the MEG analysis, and all the other people, and especially the deaf people that also help us getting in touch with other potential volunteers. And I thank you all also for the attention.
I had some questions about the behavioral relationship, some of which might just be things I missed. But so I know I heard you say, or I think I heard you say that there was a correlation in the voice area for the deaf, but not the control groups with behavior. But what I wasn't sure if you said if there was a main effect difference on this measure of face recognition.
Yes, there was.
So the deaf subjects were...
The deaf are better than the control groups. So you see this like plot on the...
But not better, the controls
not
are
better than the hearing signers.
The hearing signers, right. So, but the hearing signers didn't show this relationship?
No.
No, okay. So my question was in... areas where the control groups do show a response, that is not the voice area, but part of the face network, whether you see behavior relationships there.
We haven't looked at that yet. No.
And is it a range restrict? Oh, you haven't looked at it? So,
no,
okay.
I haven't, no.
Okay. And in general, in the literature, are the magnitude of univariate increases in face areas correlated with face recognition performance? I hadn't heard that before.
That I know, I don't think this is being like investigated in the hearing signers, so I don't think to remember that there is a particular study on hearing signer that shows like higher performances on face recognition tasks or lip reading, for example, and in particular. So
I guess I'm curious to know your thoughts about why you even went looking for this relationship, and given that you found it and in the absence of a group difference between the deaf and the signing control group, right?
Well, I still see this in the control group, right? So there is a significant difference between the deaf and the hearing control. And the hearing signers, this is a very interesting question. I think, I think this is also like pointing to the fact that I need to look into this, because the hearing signers, the idea is that the only way they differ from deaf people is that they can use sign language, but they hear. But in fact, in this sample, and I think it's very common when you're looking for these people, you find people that were born from deaf parents. So they basically spend part of their really like early phases of their development using a lot of visual information. And I think this might actually be explaining more why we see this like very, let's say, good ability to look at faces, recognize faces. It might be something that has to do with the fact that they are sensitive to some sort of the visual experience they have, but their like auditory cortex can still receive like the acoustic stimulation. So.
Right. It just seems like you're giving a different explanation. Here you want the explanation to be the recruitment of this other brain area.
Sorry, and for the sign. Could you repeat? I couldn't hear well.
We can talk about it afterward. It's okay. I don't want to take other people's time.
Okay, one short question, perhaps. Okay, thank you very much.
You're welcome.
Next speaker is Markus van Ackeren.
Okay, so first of all, I would also like to take this opportunity to thank the organizers for this fantastic opportunity to share my work with you in this format. Okay, so as we know, when a person is born blind, a person born without sight, the areas that are typically involved in visual processing, areas in occipital cortex, start to reorganize and become more sensitive to processing information from other sensory modalities, like, in fact, audition or touch. And this makes the blind actually an excellent model to study to what extent the functional organization of the occipital cortex is shaped by visual experience. Now, actually, it turns out that much of the functional specialization in occipital cortex is more or less retained in, at least then sensitive to other sensory modalities, but the functional organization is similar in the early blind versus the sighted. And one example is the fact that, for example, dorsal occipital-parietal pathways in the blind respond to spatial information extracted from sounds, very much like what we do with visual information, what we see with visual information in the sighted. However, there is one example that goes completely against this idea, and that's the example of language. So as Marina Bettini and others have demonstrated, in early blind individuals, even at the lowest level of the occipital hierarchy, we see a sensitivity to speech at an abstract level that we don't usually, or that we don't observe at all in sighted individuals. And that's interesting because it relates to a hypothesis that has been formulated by Büchel in 2003 in Nature Neuroscience, and that's the idea that what we observe in the blind might actually be a reversal of the occipital hierarchy that we see in sighted individuals. So basically, rather than information projecting upwards from V1 into dorsal and ventral streams, what we see in the blind is that V1, so the earliest level of the occipital hierarchy, is actually a convergence zone of this information. Now, this is actually not an uncontroversial theory because it's very much in contrast to recent evidence suggesting that the functional architecture of occipital cortex in sighted individuals and congenitally blind is actually very much retained. So based on this debate, we formulated the following two questions that we tried to address with our research. So one was, what level of language processing is represented in occipital cortex of the blind, and to what extent is it still sensory-based? And the second question was, where in the occipital hierarchy of early blind individuals is this information actually processed? And so to address this question, we decided to use the technique that can be roughly classified as speech tracking. So speech tracking rests on the notion that much of the information that we encounter in speech is actually encoded in energy fluctuations in the speech signal, energy fluctuations that we can describe with the amplitude envelope that you see in red in this figure. And so, for example, what we see is actually that most of the energy in this temporal envelope of speech is basically centered between 4 and 8 hertz, and this happens to be also the temporal scale at which we see syllable transitions in speech. Now, what we can do with this envelope is we can correlate or calculate the coherence between these energy fluctuations as characterized by the amplitude envelope, and we can relate it directly to the signal we can measure with time-sensitive measures like MEG or EG, right? And so we can demonstrate that neuronal populations in auditory cortex actually align to these temporal dynamics of speech. While that might not be very surprising in itself, what is interesting is that as we move away from these primary early auditory areas, we see that although general acoustic sensitivity decreases, we see that at some point the tracking of neuronal populations of the speech envelope actually becomes sensitive to what is being said. So when we compare a situation where we have a story or some kind of intelligible speech, and we compare it to a different condition where we have just the same or similar kind of energy fluctuations, we see that in these sort of more peripheral areas, we see enhanced speech tracking when we can extract some kind of meaning from the utterance. And we think this, and actually many others too, that this represents a kind of interface between the acoustic processing on the one hand and the language network proper on the other hand. And so we thought that this might be an interesting framework to test to what extent actually these occipital cortex in the blind is, or language processing in occipital cortex of the blind is still tied to the sensory input modality. So we tested a group of early blind individuals and a group of sighted controls matched for age, and we put them in the MEG, and we presented them with about 1-minute segments of famous international audiobooks like Pippi Longstocking or Voltaire. And we, in addition to that, we used 2 levels of spectral degradation to gradually impair intelligibility. And I'm going to talk about that in a second. And to test comprehension, we used validity judgment. So, for example, people had to indicate whether there was a queen in the story. So let me just say one word or a few words about the way we degraded the stimuli. So basically what we did is we used a technique called channel vocoding. And the idea here is that we take the envelope, so we take this characterization of the energy perturbations in the signal, and we fill it with bandpass filtered noise. And so this is what you see basically in the second row here. So you see that while we keep the amplitude envelope actually more or less the same in the 3 different conditions, we gradually distort the fine spectral detail. And so the effect of that is that we can actually distort it quite a bit. So even if we just filter the signal into 8 different frequency bands, intelligibility doesn't suffer at all. At this level, it's highly degraded. It would be difficult to say whether it's a male or a female speaker, for example, but intelligibility doesn't suffer. Whereas in the one-channel vocoded condition, it's impossible to understand. But still, like, the envelope is the same across conditions. So before I go into the results from the blind, I just want to give you, because you might not be that familiar with this kind of signal, just a general idea of what kind of data we can expect from this cerebroacoustic coherence analysis. Okay, and for this, I just collapse all the groups together and all the conditions. And I show you the coherence spectrum from left and right temporal sensors in MEG. And so this basically, roughly, you can interpret as the relationship between the amplitude envelope of the speech signal of these stories and the MEG signal on the other hand. And what you see is, like, there's a marked peak in left and right temporal sensors, actually exactly at this point between 4 and 8 hertz, where we see the most energy also in our speech envelope, roughly corresponding to these syllabic transitions. And you can probably tell from the topography, but if we then go into do source analysis and we look into source, we see that also the peak, so the area where the acoustic envelope is most aligned to the neuronal populations, is actually in early auditory cortex. Okay, so next again, keeping the groups together for now, we divided up our different conditions, and we wanted to know what part of the cortex tracks information from the speech envelope more strongly if speech becomes intelligible, right? And so what we found here is that this region, not in primary auditory cortex, but more like MTG, STG, just in the periphery, where we see sort of a gradual decline in cerebroacoustic coherence as the conditions become less intelligible over time. So at this point, let me take away one question already. So you might wonder, why do you find it on the right side, whereas language is usually represented on the left side, as we all know, right? Actually, I think this illustrates the beauty of this method, because remember that we're not looking at language processing proper, but we're looking at the interface between acoustic processing and up to the moment where it just enters the language system, right? And actually, there's theories like the one from David Purple arguing that at that level of acoustic processing, there is a division of labor between left and right auditory networks, where high-frequency information, phonetic information, is encoded on the left side, whereas information at lower frequencies, so the kind of information we're looking at the syllabic rate, prosody, is more represented on the right side. Right, so that's basically why we don't find the classic network, language network here, and this is also very much expected. Okay, so now finally I will go to the early blind, and so the first question we asked here, again independent of the different conditions, we just asked, okay, are there regions in the blind brain that align to the acoustic features in the signal more strongly than in the sighted? And if we do that, what we find is this parieto-occipital network, where we find reliable speech tracking in blind individuals, so where neuron populations align to these temporal features of speech, but not in sighted individuals. Okay, but the main question was, so which part of this network or is there a part of this network that also shows sensitivity to speech, where this sort of, these temporal dynamics are modulated by intelligibility? And so what we find is that really speech tracking increases during intelligible speech really only at the lowest level of this hierarchy. So it's really centered on right calculi and sulcus that we see this sort of enhanced speech tracking modulated by intelligibility. And so just to like define this effect a little bit more in more detail. So when we look at cerebroacoustic coherence in precuneus, cuneus, we find sort of a more general difference, more or less independent of the conditions in early blind versus sighted controls. Whereas in calculi and sulcus, we see that it's really the two intelligible conditions that drive this effect. And one thing to notice here is that, so you will have guessed that we've also applied a low-level manipulation to the signal in a way, because we've distorted the fine temporal structure. But you see that this is actually not what causes the difference here. It's really the intelligibility of these stories that drive the effect. Okay, so just to wrap up, so we've seen that parieto-occipital areas in the early blind track acoustic information as what we see in speech, reliable and more strongly than in sighted. But it's really only at the lowest level of the occipital hierarchy that we see enhanced speech tracking in response to intelligible speech. So it's really like, yeah, it's really just at the lowest level of this hierarchy in calculi and sulcus. And so what we think we have here is evidence that for this sort of gradient from parieto-occipital cortex to the lowest level of occipital cortex, this inverted hierarchy that Büchel proposed in 2003 in early blind individuals. Okay, so finally I would just like to thank the other members of the lab that have been involved in this project at various stages. And if you have a more detailed question, I also have a poster, so feel free to drop by. Thank you.
Any questions? Need a bit of time to digest.
Mm-hmm.
Thanks for a great talk. So in you didn't play the stimulus samples, so unfortunately I'm not sure exactly what is left in the intelligible conditions and what is removed, but my sense is that probably more than semantics is removed in the unintelligible versions. So yes. Maybe things, I mean, various aspects of even phonological regularities in the language, aspects of grammar, syntax, that kind of thing. Do you have an idea moving forward of kind of what you think might be going on in this like low, high level early occipital area, and how you might test it for what specifically it's representing?
Yeah, I mean, in a way, at this level, we're still kind of at the level of acoustic processing, right? So like, yeah. So I mean, one thing we thought about like to go for next is actually like to look at the connectivity with these regions, to see whether actually like we see this sort of relationship with the language network or with particular parts of the language network at different scales now that we have this gradient. In a way, the metric that we're looking at, it's sensory based, right? And that was our main question. So when it comes to like different, like let's say syntactic differences, phonological differences, actually phonological difference, I'll talk about that in a second, that might still work, but I mean syntactic differences, semantic difference, it will become more difficult with this method. The main focus was really to test to what extent it's still sensory based. With the phonological part, it's actually quite interesting because there was this recent paper, well, recent, it's 2013, by Groß. I mean, there is a couple of ways to address that. One is obviously with the new experiment with TMS. There are also ways with our data actually to see, well, we haven't done it yet, but to see whether there is sort of a, let's say, a dominant information flux at different points in time between sort of this low-level visual area, occipital areas, and auditory cortex. So, I mean, this is something we can implement in the connectivity analysis, but ultimately, something like TMS, for example, would be a very nice way to also see whether you can actually perturb that system. And I think there is also evidence from other domains where we can show that actually, like, zapping these areas in the blind impairs performance but not in the blind.
Thank you.
Sighted, sorry.
Just a quick question. Is there any evidence of where in calcarine cortex these effects are centered?
Where the effect is centered?
In early calcarine cortex, in V1.
Yeah, so I mean, it's right lateralized, but so at this point I have to draw the MEG card. So the thing is, it's not fMRI, so we're dealing with like 10 to 50 millimeters full width half maximum, and yeah, I mean the peak is.
Yeah, the reason I ask is because, as you know, there are direct connections now. I mean, we know from A1 to V1, and it's peripheral. So whether that was co-opting the same kind of pathway to do this was the question.
It's very popular. It's a very good question, actually. No, okay. Well, I think in general, I think this is very much in line with what we've seen with Alfonso Caramazza's talk, actually, that like somehow this area V1 is really the one that shows the most drastic changes in the early blind as compared to our sighted controls. So I think that's somehow congruent.
Okay, thank you very much, and thanks everybody. We'll be back in 1 hour.